UCSF. So, fun fact about Dave. We talked on the phone for about 2 years and wrote 2 grants together before we actually met each other in person. But it has been a delightful experience the entire time. And so he's going to talk today about germline transgenic approaches in prairie voles.
Just while I'm getting plugged in the spirit of actually making this as interactive as possible, I just want to really encourage interruptions as much as possible during the context of the talk. We will have actually, ideally 10 minutes or more to really go through and have sort of an open discussion about some of the technologies that we'll be talking about. But this is very... just want to encourage that. Also remember that there are note cards in the back. So I just want to thank you guys all for coming, first of all. And thanks Steve and Zoe for kind of putting this all together. Steve for facilitating this. Just want to start by sort of talking to the community in general and saying that what this talk is really sort of geared towards is to sort of bring everybody up to speed on some of the molecular technologies that are collaborative branch, but I think also the field in general is really sort of itching, if not aching, to really incorporate into the biology of voles. So I'm not going to be talking a lot at all about the biology of voles because I think we're all here for the same reason. And I'm going to just give you the punchline right now, at least for this part of the grand end talk, which is that CRISPR in voles works, and it works very well. And so for those of you that know what that means, go to sleep. We'll wake you up in about 20 minutes. For those of you that want to know what that means, that's what I'm going to be spending the rest of my time kind of talking about it. There we go. Okay, so what is CRISPR and how does the system work? And so I'm going to be talking about this in a couple of different levels and using a couple of different approaches for different purposes. So the CRISPR-Cas9 system is really a directed system for introducing double-stranded DNA breaks. And so I'm not going to go into the entire history of the field. Most of you know how contentious and how sometimes entertaining it can be. But suffice it to say that the CRISPR system, the Cas9 system, is really comprised of a ribonucleocomplex. And so it's the Cas9 nuclease, which is a nuclease that binds to a structural as well as a targeting RNA. And in some cases, this is a 2-part system, which is how it exists in nature, in bacteria. This has also been adapted to be a 1-part RNA, which allows us to very efficiently sort of modularly manipulate this system. So the Cas9 nuclease basically is, as I mentioned, a double-stranded DNA nuclease that cuts both strands of DNA in a targeted manner. The targeting is achieved by what's called a guide RNA. And so what this allows us to do is really target the Cas9 enzyme to any part of a given genome and introduce double-stranded breaks. Now, double-stranded breaks by themselves can be extremely useful in the context of mutagenesis, which is what I'm going to be spending most of our time talking about. When a double-stranded break occurs, various processes that are normally part of the DNA repair mechanism can facilitate the introduction of mutations into a given region of the genome. That occurs either via, for example, non-homologous end joining, which allows the introduction of small insertions or deletions at the target site. Now, the target site is specified by about 20 to 25 base pairs that are part of this guide RNA. And so that allows us to really, again, hone in with the Cas9 enzyme on any region of the genome, introduce a double-stranded break, and let nature sort of do its job. In addition, one of the sort of next steps that really allows us to generate some of the transgenic technology that a lot of people, again, have been talking about and are really looking forward to, incorporates homology-directed repair. So again, capitalizing upon the endogenous repair mechanisms of the cell to introduce a donor DNA construct into the location where a targeted break is introduced. Okay, so what I'm going to start focusing on is our approaches to sort of optimize, or develop and optimize mutagenesis using the CRISPR-Cas9 system in voles. So you guys are all familiar with probably the fact that voles, unlike mice, for example, are induced ovulators. And so what we had to do is really sort of develop a system by which we could really efficiently harvest vole embryos for manipulation, and then manipulate these embryos in vitro, and then introduce them back into a pregnant recipient so that we could actually generate transgenic animals. And so one of the things that is probably more relevant to the people that aren't necessarily only working on voles in this audience, is that as we were optimizing these techniques, we sort of identified, along with originally with the, sort of the stem cell field in general, that the conditions for sort of embryo manipulation, embryo culture, for every species seem to be bespoke. There are differences that are subtle but really important in terms of how you need to propagate embryos and cells to really facilitate both pluripotency, if we're talking about embryonic stem cells, which we're not going to be talking about today, or to actually even just culture an embryo so that you can generate a live animal. So what I'm going to do is walk in a little bit more detail through the protocol that we've sort of developed, and again, please interrupt at any point in time if you have questions about this process. So in general, we have found that variables are not amenable to superovulation techniques. So these are hormonal manipulations that, for example, in mice, can increase the rate and the yield of embryos harvested from a induced female by almost tenfold in some cases. So what that has led us to sort of adopt is a system of paired matings, whereby we use about 40 pairs of animals to generate about 100 to 120 embryos that we can then inject. So what we do is we pair the animals overnight. So these are sexually naive animals that are basically same-sex housed. We put them together overnight to begin sort of the induction of estrus process. After one night, we put in a divider. This divider is both transparent as well as has holes so that the animals can at least interact nose to nose. They can certainly smell each other. And what this allows us to do is, for example, if we take 40 pairs of animals, to synchronize the females in terms of where they are in their estrus cycle. So that goes for another day, approximately. And then, depending upon the stage of the embryo that we want to manipulate, so in the case that we're talking about with CRISPR mutagenesis, we're talking about a single cell embryo, which is basically 18 to 24 hours after fertilization. We then pull the divider, depending upon on the next day, when we want to begin harvesting embryos. So we pull the divider, and then 18 to 24 hours later, sac the females. From the females, we then dissect the oviducts, and either through flushing or through just simple shredding of the oviducts, we harvest single-cell embryos. Okay. These embryos are then put into culture and injected with a mix of Cas9 protein. Also, we use Cas9 mRNA, which we found that increases the efficiency, as well as the guide RNAs, which allow us then to target any gene that we're interested in for immunogenesis. We go through a round of microinjection, and this is basically the same technique that's used in mice and rats. And so I'm not going to talk about that in excruciating detail, but again, these are some of the techniques and resources that we're interested in sort of sharing and actually training members of the community if they're interested. So one of the things that we do that also allows us sort of a quality control for any approach and targeting any gene is rather than introducing these single-cell embryos back into an animal immediately after injection, we actually culture them to the blastocyst stage in vitro. This goes for about 4 days. What that allows us to do is, first of all, just tell, did our injection work in the sense that did we put something in that didn't kill all of the embryos, but rather allow them to grow regardless of sort of the gene that we're targeting. And also, it allows us sort of as a quality control metric to take the blastocysts that really look healthy, right? If something looks apoptotic, if something looks just generally unhealthy, we generally don't want to bother transferring that to a recipient female. Now, one thing to sort of note is at the same time that we do the injections, we then set up a separate pair of animals, in this case a vasectomized male, that is then paired with a female to induce that female into estrus herself, but as a recipient for the embryos that we're manipulating. Okay? So after we culture the blastocyst, we then take these recipient females, the pseudo-pregnant females that have been induced into estrus, and then transfer these blastocysts via surgery into the uterine horns. So again, this bypasses the oviduct. These uterine horns, these uteri, are actually receptive for implantation. And so this allows us to, again, really optimize for the number of embryos and transgenic animals that we actually end up generating. Okay. Let them go for about 3 weeks, the gestational period of the vole, and then we get G0 animals. Now, one of the important things to remember is, even though we are injecting at the single cell stage, so technically there is basically only one copy of the full genome, we do find chimeras at a level that's much, much higher than what's reported in mice. So what that means is that a couple of things are happening. First of all, the mutagenesis induced by the Cas9 system is probably happening maybe after a cell division, or a couple of cell divisions. That also leads us to a certain problem that we've sort of begun to optimize over, which is that this animal is a chimera. Parts of its genome are untouched, or parts of its body has genomes which are untouched, and parts of it are not. And so what we do in order to make sure that we haven't missed animals that, for example, are wild type in their tail, but mutant in their germline, is to cross every G0 and screen about 15 F1s. So that's about 3 or 4 litters, let's say. And we then can basically screen through enough animals to say, okay, if we don't find one in the first about 15 animals, we've basically sort of said, okay, this animal isn't a mutant. It doesn't carry any level of chimerism, at least certainly not any chimerism that can be passed on through the germline. So let me just go through the numbers now. As I mentioned, we start with about 40 pairs. That gives us about 100 to 120 embryos. We inject those embryos, and then from those probably get about 40 to 50 blastocysts that are sort of viable and healthy by our standard. About, I would say, between 15 and 20 of those blasts are transferred to a given recipient female. So we usually do about 3 recipient females from a given batch of embryos that we inject. From those 3 females, we get about 2 to 3 animals from each when they actually implant that are our G0s. And I can say now for a handful of genes, every gene that we've targeted successfully, in about 2 rounds of injections, we can obtain anywhere from 3 to 6 alleles of a given gene. And this is just for mutagenesis now, right? I'm not talking about homology-directed repair or knock-ins per se. But what that gives us is an allelic series in some cases. And remember, these are random mutagenesis events. So some of these are insertions, some of these are deletions, and some of them actually are in-frame mutations that actually result in presumably partial loss of functional alleles. But suffice it to say, this protocol works very efficiently, as I mentioned, for the number of genes that we've targeted. Okay? So in terms of where the field is, in terms of thinking about technologies that you guys are sort of talking about and asking about, this is something now that I think is very, very certainly portable, as well as easily doable. And so that's one of the things that I think that's a goal of this meeting, is to really sort of develop a system by which the community in general can use these techniques, but also start to think about how do we want to centralize not only the techniques, but also the resources that are developed, so that individuals don't all have to, again, reinvent the wheel, or in this case, the Vole. So just proof of principle. The first gene that we targeted, not surprisingly for this obvious sense, was the oxytocin receptor. And with our first round of injection, we basically obtained 2 alleles. So this is using a single guide RNA. The important thing that I forgot to mention when I was talking about the Cas9 system is that where you target with the guide RNA, is restricted only by a 3 base pair sequence. This sequence is the PAM sequence, which is species-specific in terms of the bacteria that the Cas9 protein comes from. The majority of techniques that we're going to be talking about and that have been developed in the field use the Cas9 protein from pyogenes. The PAM sequence in this case is the famous NGG, so any nucleotide followed by 2 Gs. And this allows the holoenzyme to actually target the region based off of homology with the guide RNA. So we, using a single guide RNA, obtain 2 mutations in the oxytocin receptor, both of which are presumable loss of functional alleles. One is a single base pair insertion that creates a premature stop codon that gives rise to an 84 out of the 388 amino acids of the peptide. And another is what is presumably a 6 base pair deletion and a single base pair insertion, so a functional 5 base pair deletion, which gives rise to an 82 amino acid truncated protein. Just as validation, when we look at our first allele, we see that we completely abolish oxytocin agonist binding in the Vole brain. So again, this is a validated loss of function allele. And sort of an interesting correlate for obviously all of the Vole people in the audience is that in the absence of oxytocin receptor, we don't really see binding of at least this ligand, suggesting that it's not binding to any of the other receptors that many of us also care about. Okay, So this is kind of where the state of the field is in terms of mutagenesis. The next step then is how do we actually start to generate knock-in alleles to do all of the manipulations that we're used to seeing in other species, in particular mice and rats. And again, this relies upon homology-directed repair to introduce, via the double-stranded DNA break, a transgene from a donor construct. So in the mouse literature, a number of approaches have been tested and in theory optimized to really facilitate and optimize homology-directed integration of donor DNA constructs. The first is what was sort of pioneered off of simple homologous recombination, which, for example, exists in embryonic stem cells, which is homology-directed repair following, homologous recombination following the introduction of double-stranded DNA break. And so in this case, this uses longer arms, so these are now regions that are homologous to the flanking sites on either side of the double-stranded DNA break that flank a transgene. Following introduction of the double-stranded DNA break, homologous recombination facilitates the incorporation of this transgene into the target site. Now, one of the important things to always remember is that when you're designing DNA constructs for homology-directed incorporation, that the CRISPR sites, the guide RNA binding sites, need to be not present in the donor. Otherwise, you're just going to simply cut your donor sequence itself. And this is something that can be sort of capitalized upon in terms of making sure that you're integrating into the right site or in the right direction. But for at least the first pass, we basically are designing all of the constructs that we're using to simply lack the target site itself. Now, sort of using the sort of techniques that CRISPR itself capitalizes upon for mutagenesis, or in this case, non-homology end joining, the simple introduction of a transgene with the flanking sites of the guide RNA can also lead to some level of repair via error-prone mechanisms. So this is just incorporation of that as the sort of repair mechanisms are targeting the site of the double-stranded DNA break. This is in general considered to be and documented to be significantly lower in terms of its efficiency. And so this is not something that we're really approaching. Now in mice, which have really sort of been optimized for all of the techniques that we have sort of started to test in Vole, you can also use microhomology mediated end joining. And so this is using flanking sequences as short as 20 base pairs to introduce transgenes, again, into the site of a double-stranded DNA break. Again, the molecular mechanisms are a little bit less important. One of the things that is a limitation of the microhomology is the size of the transgene seems to be very, very restricted when you're using short flanking regions. And so, for example, the introduction of a lock site, this may be facilitated by microhomology-mediated repair. But for larger transgenes like Cree or other reporters, things like that, it seems to be that those are a little bit too large to be incorporated, again, with microhomology. And so really the technique that we've sort of really started to optimize in the lab is to use a combination of the principles of homologous recombination with homologous end joining. And that is to use large flanking sequences in the context of a DNA donor that doesn't involve the sort of plasmid backbone that is typically used for homologous recombination by itself. And so what you have is basically just in this case, double-stranded DNA donor that has only the flanking sequences and then the transgene of interest incorporated or injected with the ribonucleotide complex, and then reassess for incorporation into the locus of interest. So in order to sort of optimize this system, we are taking advantage of a paper that came out about 2 years ago, which basically demonstrated that you can use a given target locus, in this case the actin locus, to really optimize for homologous recombination or incorporation in vitro as opposed to waiting for a transgenic animal. And so this is basically using a guide RNA that targets the final codon of the actin protein and incorporates a P2A, so a fusion peptide, followed by an M cherry. And so what this allows us to do is, following injection, to really just screen in blastocysts as opposed to having to wait or screen through transgenic animals after they've been born. And so Ruchira Sharma, who's the postdoc in the lab, who's really pioneered all of this work, has really sort of started to use the system to optimize the techniques for knock-ins in voles. And again, what we're screening through at this point in time is for red embryos as opposed to red animals. And so this is now kind of the next phase of where we are in terms of the technology of the grant. So just to sort of now transition to some of the other technology that Zoe is really going to start focusing on, the Cas9 system has a lot of flexibility, which most of you are probably exhaustively familiar with. Because the Cas9 riboprotein complex really allows you to target the Cas9 protein heavy region of the genome, you can then just use the Cas9 protein itself as a tether for basically any other of the effector functions that you can imagine. And in particular, what we're interested in, and I think most of the people in this room are interested in, is gene regulation. Now, as I mentioned, the Cas9 itself, protein itself, is a nuclease that induces double-stranded DNA breaks. However, it has very, very specific nuclease domains for both strands that it actually induces a break in. And so these have been actually mapped and mutated to create a dead Cas9 protein. So this is now a functional protein that binds to a guide RNA complex, but has no nucleus activity. And so what this allows us to do is now use this as a scaffold to affect gene regulation based off of where we target a given gene. So you'll see the abbreviation dCas9. This is going to be dead Cas9. And this can now be used in the context of gene regulation in a number of different ways. So first of all, for inhibition, one of the more canonical systems that has been used is the dCas9 coupled to a CRAB domain, which is the transcription inhibitory domain. Targeting, for example, the transcription start site of a given gene allows you to repress its transcription. Part of this is thought to be simply through steric hindrance. The complex sitting down at the transcription start site basically prevents RNA polymerase from running through and inducing, and actually transcribing the gene downstream. And more importantly, we can use various transactivating domains coupled to the Cas9 protein to induce gene expression. And Zoe's going to talk about this in more detail, but depending upon the sort of scaffold mechanism that you use, either a direct transactivating domain, for example, the PP64, or other transactivating mechanisms, we can use those to then positively regulate gene expression. And again, I'm not going to go through this in exhaustive detail. So I'm going to stop here for questions on the CRISPR-Cas9 system.
What's the advantage of using the CRAB to repress expression versus just using the CRISPR to mutate the gene?
So there are a couple of different reasons that gene regulation may actually be a more useful technique. One is the efficiency of somatic mutation. So if you're targeting a given region of the brain, is much lower than if you're simply expressing something that can directly knock down or inhibit expression. And so the chimerism that you actually get, for example, if you put in a virus to mutate genes in particular in neurons being postmitotic, is much, much lower versus just actually putting in a protein that sits at the locus in all of the neurons that are infected to then inhibit gene expression. So that's the principal advantage that you actually get. And then the other thing is the regulatability, right? You can imagine that if you have a inducible system, you can actually regulate gene expression using an inducible inhibitory CRISPR versus something that's a permanent mutation. Right?
Just a functional question. When you're basically doing IVF on your voles, do you ever freeze the blastocysts after you've created them?
So we haven't frozen blastocysts. Again, as I mentioned, where we have really worked on cryopreservation is with ES cells. And embryo preservation in general in rodents is pretty dismal. You can freeze embryos, but it's a little bit harder, and most people tend to freeze them at earlier stages than the blastocyst stage. We haven't really worked on trying to optimize that for voles. But that's definitely something that thinking about what are the resources that the community needs in general, is it a Jackson Lab sort of model, or is it, a different one in which we can actually just freeze down embryos of a given transgenic.
Do you have any method to look at off targets for this CRISPR?
So that's actually the second part. Is
Oh,
we
yes.
no, not specifically for off targets, but we have again, I'm not going to necessarily go into a lot of detail, but we've sort of started to begin to resequence the prairie vole genome to get high resolution sequencing for prairie as well as the meadow, and that's work that's pioneered by Kristen Berenzon, in the lab. But what we do for off-target events is we actually just outcross 7 generations to really sort of isolate the mutations of interest.
But for the explosive receptor, we don't.
Yeah, so what we basically...
I didn't hear the question.
The question was, how do we deal with off-target events? So, again, one thing that I didn't mention about the biology of CRISPR is that while it is sort of specific and specified by the target sequence, you can imagine that there is some redundancy both in terms of biological noise as well as sequences in general. And so in the absence of a very high resolution genome, you may actually design a guide RNA that hits a couple of different places in the genome, and so therefore what you actually get mutations in may not only be the gene of interest. What people in both the mouse field as well as now in rats do is once they have a mutant animal, they simply just sequence with high resolution to see, okay, are there other sequences that have been hit? And you can obviously optimize computationally for sequences that have some level of homology to your target sequence for your guide RNA. Because we don't have that level of resolution for the Vole genome, what we've done, for example, with the oxytocin receptor, is to outcross, right? And so if you think about the way genetics works for outcrossing is you basically, every generation, have 2 to the n in terms of the level of the background genome versus your original potential mutation. And so it's sort of considered after 7 generations that you basically reduce to less than 1% the likelihood that something that isn't directly linked at a chromosomal level is responsible for the phenotypes that you're observing.
So, just previously talking about off-target sequences, what about like region-specific sequences? Are you able to do CRISPR-Cas9? From what I saw earlier, you're able to globally knock out oxytocin, but what about specific regions instead of just a whole global thing of
Right, so Zoe's going to talk in a lot more detail about the sort of approaches that we can use to at least knock down, if not knock out, like Larry was asking, gene expression in individual regions. Part of where that combination then comes into play is when you have conditional alleles. So, as I mentioned, not only generating reporter lines, for example, a Cree line for a given gene, but then conditional alleles where you can flox out using directed insertion of lots B sites. Okay, so for the sake of time, what I'm just going to do very quickly is kind of go through sort of a separate set of resources that my lab has been working on in collaboration with 2 different labs, Jessica Tolkien in Cold Spring Harbor and Mike Schatz at Johns Hopkins. And this is basically genomic resources for voles, in particular prairie voles and meadow voles. So the idea being to do at sort of a transcriptomic or genomic level, many of the comparative studies that all of us are interested in, sort of the field in general, was originally built upon. So the current prairie vole reference genome is quite exhaustive. But the original sequence from Broad had long scaffolds that had many, many regions of non-specific sequence. So out of the about 2,000 megabases of the prairie vole genome, about 190 are really missing from the current assembly. And that includes many of the interesting regulatory regions for some of the genes that all of us are interested in, as well as interesting genes themselves. And so what we sought to do was to resequence the prairie vole genome using more recent techniques, as well as sort of coupling upon, or piggybacking upon multiple approaches to develop a more high resolution and complete assembly of the prairie vole genome. And so what we started with was packed biosequences, which give us long reads. And then using, again, without going into excruciating detail, a series of computational steps to assemble the PacBio genome, as well as coupling this with bio-nano sequencing, which allows us to get even longer scaffolds. We've now sort of developed a much higher resolution and more complete version of the prairie vole genome that, at least at an individual level, can become accessible. And obviously we're interested in sort of presenting this to the community in general once it's basically complete. In addition, the annotations we've redone using transcriptomic profiling from a number of different regions, the brain, the liver, the testes, as well as embryos. And so we now have sort of a wide range of transcripts that we can then use to annotate the genome. This has been facilitated first by comparing these sequences to the current assembly that exists. And so what you can see here is that using these approaches, we've actually improved the reference genome quite considerably. I'm not going to go through these metrics in detail for the sake of time, but suffice it to say that we have regions that previously had been uncovered, as well as better annotation and better resolution for many of the regions that we're interested in. And so the important metric, the N50, for those of you that are aficionados, that is one of the metrics for the quality of a genome assembly. We've improved that considerably using these approaches. And so now, in addition to the prairie vole genome, we are in the process of making an assembly as well as annotation of the meadow vole genome. As I mentioned, we had the original prairie vole genome as a reference base for our assembly, which facilitated it significantly. And so the question that we're sort of dealing with now at a sort of computational level, a theoretical level, is whether or not we are going to try to do this as a complete de novo assembly, or use the prairie vole genome as a reference for meadow. And obviously there are pluses and minuses to both of those approaches. We'll probably end up doing this both and see what we end up getting. But again, this is a resource that we're very interested in making available as soon as it's actually tractable to the community at large. And so, again, just for the sake of time, I'm going to stop there. I really want to recognize Ruchira and Kristen, who are here, so please inundate them with all the questions that you have. And thank the many collaborators that we've had, in particular in my lab. I also want to recognize Karen Bales, who sort of originally gave us our voles, as well as Manoli Sperry, who gave us the meadow voles, gave us prairie voles in our meadow voles. And members of Karen's lab who really helped sort of in the technique and protocol development. A technician when I was a postdoc in the lab for Hasham, or Mari Cruz, who also helped sort of with the initial development of these protocols, as well as our collaborators and you guys.
Just want to reiterate, trainees first. If you have a question, we'll get you the mic.
Go forth and CRISPR.
All right, nobody, Larry had a question.
When you're working with transgenic mice, you know, you can breed siblings together. One of the things about voles is that you have to keep them sort of outbred, and we found that that's really important. So have you thought about, I mean, how do you deal with that with the transgenics, you know, because you need to maintain that genotype. Like, you basically have cross animals that are related,
Yeah.
but you also want to increase the genetic diversity.
So what we have done is with every mutation that we have, we take independent, so we, especially when we're doing the outcrossing, we do for each mutation independent outcrosses. For the outcross themselves, we actually alternate whether we use a male or a female for each generation so that we don't get effects of the mutation being transmitted either epigenetically or at least environmentally. And so by doing that, we basically cross them into a common but distinct genetic background that we can then use to bring those animals together to get homozygotes. So every line that we have basically two independent families that we can then use to sort of create crosses to do the genetics. And that is something that so far we haven't developed a way. I mean, presumably, you know, we can find a gene that actually is responsible for the incest avoidance and then bypass that sort of mechanism, but that's its own question. Yeah, we haven't found that.
So if you're affecting genes that potentially affect parental behavior, shouldn't you also be doing cross-fostering?
So that is another option. So far, the genes that we have been looking at, the one that I am thinking about that probably hasn't for parenting phenotype in hens, we haven't seen significant effects in heterozygotes, which has been a boon. But in particular, some of the genes, for example, those from clinical literature, do seem to have heterozygous phenotypes, both in clinical populations, but then also in voles. And so there, what we've sort of done to sort of bypass that is to actually, again, do the alternate sex. If we don't find a parenting phenotype, for example, in one sex versus the other, in a het, then you can actually use that, obviously. So for example, I can use the example of the oxytocin receptor. Homozygous females have a significant lactation deficit. And so obviously their pups are rough, and so you wouldn't want to breed with a homozygous female. Right? So those are the sorts of things that we have to take into consideration at a gene-by-gene level. And I think something that's a little bit more going to be specific to individual mutations and whether or not you actually need to do cross-fostering. One of the things, going back to Larry's question, that we have sort of done to also get around the fact, I don't want to say get around, but sort of deal with the fact that our voles are outbred populations, right? So they have enormous genetic diversity, is when we originally got the families that we had as our founders and our starters, we actually built sort of a common genetic background line that has 8 different lines, so grandparents, and then going through crossing multiple animals from different lineages to sort of create a common genetic background for the voles that are in our colony. Obviously, in the context of where you have genetic diversity that you can then capitalize from a behavioral level, the ability to then take a given mutation, cross it into a specific background, that becomes both really interesting and very useful to think about how modifiers are operating at a genetic level. But we've tried to sort of at least normalize against that by using this common genetic background. I don't know for the purposes of CRISPR mutagenesis whether that is a hindrance. Certainly in the context of embryonic stem cell work, any potential mutations, even SNPs, can dramatically decrease the efficiency of homologous recombination if you're talking about it in ES cells. So far, in the literature, at least of CRISPR, both in other species as well as our own experience in voles, that doesn't seem to be a significant problem in terms of thinking about at least HR.
Okay, so we're gonna do a quick 30-minute coffee break so you can reinvigorate your brain with caffeine.